I'm pleased to introduce Sarah McCarthy. She is a sophomore at Stanford University studying chemistry with a biological concentration. And in her free time, she likes to cook and walk around beautiful places. So the title of her presentation is Antigen Discovery for Prevention of Gonococcal Disease.
Thank you, Carrie, for that introduction. To begin with some background, this summer we worked on developing a vaccine against gonococcal disease, or gonorrhea. Gonorrhea is a sexually transmitted disease caused by the gram-negative bacterium Neisseria gonorrhoeae. Over 78 million people contract gonococcal disease each year, and similarly to COVID-19, people with gonorrhea can be asymptomatic, which contributes to the widespread. Currently, the only treatment option for gonococcal infection is antibiotics. However, many strains of Neisseria gonorrhoeae have developed antibody resistance to antibiotics. As a result, there's an urgent need for a gonococcal vaccine since we may run out of treatment options. In order to develop a vaccine, we need to identify which Neisseria gonorrhoeae protein antigens stimulate antibody production. A modified version of these antigens could be used in a vaccine. Since there are few Neisseria gonorrhoeae protein vaccine candidates, our objective this summer was to identify new Neisseria gonorrhoeae protein antigens that elicit protective antibody responses against gonococcal disease. Our methodology began by using bioinformatics to identify protein antigens with consistent expression across several strains of Neisseria gonorrhoeae, coupled with localization in the outer membrane. We amplified our genes of interest using polymerase chain reaction, or PCR, a lab technique used to create billions of copies of a specific DNA sample. Subsequently, we performed gene cloning in E. coli to generate our Neisseria gonorrhoeae proteins of interest. Next, we utilized a protein detection technique called Western blotting in order to analyze the expression levels and solubility of our proteins. Moving on to the results, I will briefly walk through the first few figures. Table 1 shows the information about our proteins from our bioinformatic work. Figure 2 shows that we successfully amplified our genes of interest using PCR. And figure 3 shows the first restriction digest from our gene cloning process. Figure 4 shows all of the proteins that were in our samples with our specific proteins boxed in red, but what I really want to focus on is figure 5, the result from our Western blot. To orientate you, lanes 2 through 6 look at the expression levels of our proteins in E. coli, and lanes 7 through 11 look at the solubility of our proteins. Looking at the green bands in lanes 2 through 6, we can see that we successfully expressed 4 of our proteins in Neisseria and E. coli. Looking at the green bands in lanes 7 through 11, we can see that 2 of our proteins were at least partially soluble. So in conclusion, we successfully expressed 4 of our 5 proteins in E. coli, and at least 2 of our proteins were partially soluble. The next steps are to purify our proteins, immunize mice and/or rabbits with our purified proteins, and test about antibodies generated from our immunizations. I would like to thank the CHORI SSRP leadership, the Birnick Lab, and the NIH Training Grant for making this summer possible. Thank you for your time, and I can take any questions now.
Fantastic. Right on the money. All right.
That was a great presentation, Sarah. I was wondering, so I think I'd read in your abstract that gonorrhea, you don't get immunity from having the disease. And then how would antibodies or antigens be able to give you immunity if I would expect that you're exposed to all of them while you're infected? Like, what is the way that you're able to get immunity from the vaccine?
Yeah, so that's a complicated question, and we don't even, so basically with, your body doesn't remember the antigens that are inside of gonorrhea after you get the infection, and so that's why you can repeatedly get it. We focus on antigens that were expressed in at least 14 of the 15 strains of Neisseria gonorrhoeae when we were working on developing our vaccine. And so what we're hoping is then we're going to get this antigen that's expressed in a lot of different strains of Neisseria gonorrhoeae, so then the vaccine will be efficient. But that's definitely a complicated question that we are trying to address as well.
Hi, Sarah. Good job on your presentation, by the way. I saw that there was many different steps and methods to your project, including PCR and cloning. And there's many limitations right now over the summer because we aren't there physically and can't be in the lab. So I was just interested as to which parts of the methods you were most intrigued by and most excited by.
Oh, wow, that's a really great question. I was really fascinated by PCR, which is by far the most simplest lab technique that we use this summer, but I had never done it before. Last summer, I was able to do gene cloning, so I was able to do the bacterial transformation and all the moving of the liquids to get it done. So I was really excited to be able to watch PCR. My mentor set up a webcam in the lab, so twice a week I would get to observe him literally doing the lab work, so like putting the things on ice, pipetting the liquids, which was really enjoyable.